Hi everyone, welcome to my channel Pure Biology, and today's topic is that sporophytic generation, and its second part, that is the gametophytic generation. So I have already explained in my channel, so you just check out that one. So this sporophytic generation, it is the type of the plant development, or you can say the stages of one of the two stages. One stage is the sporophytic generation and another is the gametophytic generation. The two type of stages are there in the plant development, and the sporophytic generation, it has or it constitutes the two phases. One is the microsporogenesis and another is the megasporogenesis. Clear? So the sporophyte that bears the spore producing organs, and this produces the spores by the process of the sporogenesis. So sporogenesis is what? The sporogenesis is the process by which the spores are produced, and the sporophyte that containing the organs which can produce the spores. Clear? Now the first stage, that is the microsporogenesis. First one, that it is the formation of the microspores. So microsporogenesis is the process by which the microspores that are formed and that give rise to the male gametophyte, or you can say that microspores that mature to form the male gametophyte. And generally you all know that in case of flowers male reproductive part, that can contain the stamen, and stamen has two parts, that one is the filament and another is the anther. So anther is what? Anther is that of microsporophyll, or the stamen that bears the microsporangia or the pollen sacs. So anther that bears the pollen sacs. Clear? Anther is containing the pollen sacs, and its function is what? The pollen sacs function is to produce the microspores or the pollen grains. And anther generally, general typical anther that is sporangiate type, that is looking sporangiate type, or here the anther generally has the two microsporangia or the two pollen sacs, and each pollen sac that containing the archisporangia or the archisporangial cells. Clear? So this archisporangial cells function is what? The archisporangial cells generally divide in two ways, towards the outside and obviously towards the inside, and it in the outside divide to form a parietal layer, and in the inside form the primary sporogenous cells. Clear? And its division generally seen in a plane that parallel to the outer wall of the anther lobe. This is the lobe, okay? Parallel layer of the anther lobe. So its division is known as the periclinal division. Okay, this is the periclinal division. And the cells of the parietal layer, what happening to the cells of the parietal layer? The cells of the parietal layer that undergo a series of the periclinal and anticlinal divisions. Two types of divisions seen generally in horizontal division and the vertical division. So sorry, this is the horizontal division and vertical division. So the two ways named as that periclinal division and the anticlinal division. In that way, 2 to 5 concentric layers of anther wall is formed. Okay, 2 to 5 concentric layers of anther wall form, and the mature anther wall that comprises of epidermis, endothelium, and 2 or 3 middle layers. So what containing this mature anther wall? This mature anther wall that containing the epidermis, that is the outer wall, endothelium, that is the inner wall, and middle, that is 2 to 3 middle layer also seen there. Now a single layer of tapetum is also present in that. This tapetum actually composed of the single layer of cells, and this characterized by the presence of dense cytoplasm and the nucleus. So general tapetum consisting of single layered cell, and each cell containing the cytoplasm, dense cytoplasm, and obviously nucleus. Now what's the type of the tapetum? Generally two types of tapetum are seen. One is the secretory tapetum and another is the amoeboid. And its function is what? Its function is, the first one, generally it supplies the nutrients to the microspores. Second, synthesizes enzymes and that degrade or break the cellulose wall for the release of the microspores in the tetrad. And third one, that helps in the pollen wall formation. So three functions are there. First one is that first function, that is the nutrients supply, or you can say that it helps in the supply of the nutrients to the microspores. Second is the enzyme synthesis for the breaking of the cellulose wall for the release of the microspores. And the third one is that helps in the pollen wall formation. And the primary sporogenous cells that may function as microspore mother cell, or its another name is that PMC, or the pollen mother cell. So primary sporogenous cell that function as what? Microspore mother cells or the pollen mother cell, or its short form is that PMC. Sometimes may undergo meiotic division to form the new cells or to increase the number of cells before entering into the meiosis division. And here the each PMC or the pollen mother cells that gives 4 haploids in the meiosis division or meiotic division, not the mitotic division, because in case of mitotic divisions the number of cells increase, and this happening just after before entering the meiosis. So in the meiotic division, the 4 haploid cells are formed, and the arrangement of the 4 haploid cells it's known as the tetrad. Clear? Now arrangement of the microspores in the tetrad is tetrahedral. So two situations are seen. If the arrangement of the microspores in tetrad is in the form of the tetrahedral condition, then dicot will be formed, and or sometimes isobilateral arrangement is seen in the bilateral situation. So in that situation the monocots may be formed. Clear? So two types of arrangement seen. One is the tetrahedral situation and another is the isobilateral situation. And here all the microspores that remain in the tetrad, they always separated from each other. Clear? So this is the sporophytic generations. I explain only the microsporogenesis. Here I explain to you. In the next video, I will try to explain the megasporogenesis. Clear? So if you are the new person, so you just share and subscribe this channel if this is helpful to you.